Hey everyone, and welcome back to Dr. Liu's VCE Biology channel. Join me today as we explore the 2020 VCE Biology paper. We will focus on the questions covering responses to human change over time and scientific investigation. I'm sharing my understanding, but I'd appreciate your help ensuring everything is accurate. If any teachers, parents, or students notice any errors, please feel free to guide me. A student experimented to examine how light reflects from different leaves, recording the results qualitatively. Qualitative data is non-numerical information describing qualities or characteristics, often collected via observations, interviews, or surveys and comprising descriptions, categories, or labels. For this experiment, the qualitative data specifically noted the names of the different colors of the reflected light. Scientists investigated Manuka honey's effects on Staphylococcus aureus, a wound bacterium. S. aureus cultures were grown under three conditions, with Manuka honey, jellybush honey, or no honey. Option A. It states that Manuka increases the level of EF2. However, the table shows that Manuka decreases the level of EF2. Option B. It states that the ability of S. aureus to adapt to changing conditions is reduced by Manuka. This ability is related to CSPC, which the table shows is increased by Manuka. Therefore, Manuka enhances this ability, contradicting the option. Option C. It states that Manuka is more effective than jellybush at limiting S. aureus growth. The table shows that Manuka increases the average time for S. aureus cell numbers to double, compared to the jellybush. This supports the conclusion that Manuka is more effective than jellybush. Option D. It states that Manuka heals wounds faster than jellybush and no honey. However, the provided information, or table, contains no data about wound healing or recovery. To investigate Manuka honey's specific effects on S. aureus, the experiment used three conditions: Manuka honey, jellybush honey, and a group with no honey added. The no honey group acted as the baseline control. The jellybush honey group was included to compare results against another type of honey, helping to determine if observed effects were unique to Manuka or a general property of honey. This comparative setup was key to identifying Manuka honey's distinct impact, the experiment's primary objective. Dutch archaeologists discovered 12 skulls and 2 leg bones. Two key structural features of these fossils suggest they belong to Homo erectus rather than Homo sapiens: a smaller brain case and a larger brow ridge. Other accepted features include a more sloping forehead, a larger jaw, and a less slender zygomatic arch. Figure 1 depicts Homo ergaster and Homo sapiens as having diverged from a shared common ancestor, while Figure 2 illustrates Homo ergaster as a direct ancestor of Homo sapiens. This difference in representation implies a closer relationship and a more recent common ancestor in the model presented in Figure 2 compared to that in Figure 1. Such variations highlight the dynamic and evolving nature of our understanding of the evolutionary relationships between these hominin species. Homo denisovans are often absent from older evolutionary tree diagrams. Based on DNA evidence, denisovans are closely related to Homo neanderthalensis. Potential placements for denisovans on trees include alongside Neanderthals or branching from Homo heidelbergensis. Genetic studies indicate interbreeding occurred between denisovans, H. sapiens, and Neanderthals. Determining the precise evolutionary position of denisovans can be complex for students. Their relationship and timing relative to other Homo species are key challenges. Scientists do not always agree on the position of each Homo species on evolutionary trees. This is due to gaps in the fossil record, discoveries altering previous views, and incomplete or small fragments of fossils. This section, titled Human Change Over Time and Scientific Investigation, includes 3 short answer questions, worth a combined 3 marks, and 1 multiple choice question, worth 7 marks, totaling 10 marks. These 10 marks represent 8.3% of the assessment's total possible score of 120. 10 topics are covered in the assessment, split between Unit 3, 4 topics, 52%, and Unit 4, 6 topics, 47%. These topics encompass nucleic acids and proteins, DNA techniques, biochemical pathways, photosynthesis, respiration, biotechnology, immune responses, disease, population genetics, speciation, determining relatedness, human evolution, and scientific investigation. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.